And coming to the topic today, I would briefly go how and why living organisms and objects in nature manipulate light. And we discuss a little bit more about the colors in nature. And then I move on to a pigmented color with a specific example for selected molecules and polymers. Then we go into another way why the colors are produced in nature, that is a structural color, essentially optical nanostructures. And we discuss some of the details how the factors responsible for structural colors. And we go on to discuss a little bit more about photonic crystals and optical transparency. And this is intended to interdisciplinary audience, especially undergraduate and postgraduate level. Therefore, you don't need to have any background in many of this area, and it's highly simplified. So before starting, I would like to show you this video, and this video, it shows you that this video is a dynamic color change. We have a silicon wafer, we have a very thin film on a silicon wafer, and when exposed to moisture, it displays this beautiful color. And this color can be erased just by blowing some hot air, and it can be regained. And this kind of dynamic color change is very fascinating, and these are not from pigment because the material we use is completely colorless. So this actually has fascinated us that to learn a little bit more what is color, how the colors are produced, the phenomena of color. And nature actually utilizes this kind of dynamic color change, especially to overcome some threat, defense, and adapt to the environment. And why color is important? Color helps us to remember certain objects. And it often sparks our emotion, and it also can influence some of our decisions, such as purchasing. And we often have always some favorite colors. And why we are able to see different colors in the objects or animals or any other natural materials. Because human eye is capable of distinguish color in the visible portion of the spectrum. That means from UV to IR, in between those range. Unfortunately, human eye cannot perceive ultraviolet or infrared rays. But we have to remember that there are certain organisms, bees and snakes, they can perceive ultraviolet rays. And they have different anatomy because they have to adopt extreme conditions in the environment. In order to look at an object, we are also able to see the differences in the brightness and intensity. That's why if there's an image in color, it has to be presented for us in a visible portion of the spectrum and also with a varying degree of brightness or intensity. This is because of the complex architecture of the human eye, where we can have very specialized photoreceptors, and such as like cones and rods. As you can see in the electron microscopy image here, we have rod-like receptors and we have cone-like receptors. And these cones are specialized to have red, green, or blue colors. And interestingly, we have to know there are 12% of females who can also have more than 3, that's tetrachromy, like 4 receptors. So they can see more colors than ordinary people, or rest 80%. And we have to remember that there is also color blindness. So it means that red and green color is the most common, or blue and yellow colors. So therefore, sometimes we have to remember which color cannot be used when we present to a certain population. And living organisms manipulate color and manipulate light, and their way they can generate colors. And how they do it is one of these phenomena, which is called absorption, reflection, scattering, diffraction, interference, transmission, or waveguiding and lensing. And other important aspect of living materials and animals, which live under extraordinary conditions and extreme conditions, is that they are made up of all these color materials are made up of simple and ordinary materials. But the difference is that they have extraordinarily precise optical engineering. And they can utilize the limited resources in a very effective manner. It's highly efficient. And more importantly, many of these materials also have more than one function. So as I said, that living organism can manipulate light, and they manipulate light because they have certain molecules that can manipulate the interact with the light selectively. In principle, everything in nature absorbs light, but they absorb light at a specific wavelength, and without very specific wavelength absorption, there will be no color. We cannot have vision. There will be no heat, no light derived energy, or no biomass production, such as, for example, photosynthesis. And if you go to biomolecules, for example, DNA, which can strongly absorb in the UV radiation between 240 to 275 nanometer, which is specifically due to the DNA bases which have pi-pi transition. Then we come to the proteins, which have peptide bonds, depending on the type of amino acids it has made up of, they can absorb light between 180 to 280 nanometer. Once again, both proteins and DNA fall in almost in the same region. Then we have metallocomplexes, such as, for example, hemes and carotenoids. They strongly absorb. They are pigmented molecules. Importantly, water molecules absorb preferentially in the IR light. And this is why we are able to generate heat. And that's what responsible to keep our body temperature in biological tissues. I told that the DNA strongly absorbs in UV region, but that is not good. It's bad for any living organism because UV radiation can lead to DNA damage. It leads to photocross-linking of DNAs and proteins. Then how animals and we overcome this? And this is called as UV-absorbing sacrificial molecular pigments, such as carotenoids and melanin. At the cellular level, proteins, nucleic acids, and other components, they actively contribute a very broad spectrum of absorption. And then we can go more than one type of molecules, and they will allow us to achieve more than one functions by selectively absorbing light at a specific wavelength. If you look at the molecular pigment, one example that I would like to give you before I go to the structural color is the carotenoids. They have very high absorption coefficient, and also interestingly, they can function as a light harvesting system. They can also provide UV protection, and they also result in biological coloration. One interesting example is flamingos. And for example, flamingos can lose their pink color if they don't have enough dietary supplement of these pink shrimps. If they run out of this diet, their color will slowly fade. So these pigmented colors generally fades. But here is an example. It's a video taken from National Geographic. What I show you is the octopus ink. And they shoot the ink. Many of you might have seen. Octopus is famous for also the color changing, and they can change multiple color. This is the octopus that can shoot the ink, and it's a combination of many things. And here is an example. It shoots a thick smoke here. Interesting is that what is it made up of? And this is made up of a complex polymer that what we call melanin. Melanin is also responsible for many other functions, and these are like produced and synthesized in melanosomes, and it's a major component in our skin and also many other animals, sea animals like cephalopods and squids, microorganisms, retinas, and bird feather. It is also, if this is the same polymer, we can have more than one type, and nobody knows the exact composition so far of melanin. And that's why it's not possible to make a synthetic melanin with the exact composition yet. And this exhibit very broad absorption that covers wavelength from UV to visible light. This is, as I said, it's an universal absorbing pigment. It provides coloration. It provides also UV protection. By coloration means that it can also provide certain microorganisms the ability to be more virulent. It can also increase the mechanical property of certain substances, and they have a huge number of chemical moieties that can bind, for example, to various metal they help to sequester metal ions in the water. What happens in the insect, this octopus ink, is that there is a very high fraction of melanin, which is combined with the mucus, and that creates a very thick smoke. And by thick smoke, it creates a big cloud so that it can escape from the predators. So this is a defense mechanism that many of cephalopods and squids adopt. And so it's not just about the color, it's also about preventing yourself from the predators and threat and extreme environment. Now, after these two examples, I would like to move to the actual topics, the structural color. In 2005, in the Scientific American, there was a very interesting article that why male birds are more colorful than female birds. And in many birds, if you can see, it's something like peacock, for example. The male birds are much more beautiful compared to females. And they have beautiful colors and display of colors. And they can also display colors at various events. And here, for example, peacock. And peacock has this peacock eye pattern. Interestingly, many years ago, there was also research conducted in ecology department in IASC. If you remove one of these eyes from one side, then it becomes asymmetric, and then this male peacock will fail to attract a female peacock. So this is a very interesting phenomenon, that how nature adopted color to also not just for defense, but also to attract its mates. And these are called structural colors. And structural colors are observed for one of these three reasons. The phenomena such as interference, diffraction, or selective reflection of light from what we call as photonic structures. And we can see in our day-to-day life many of these kind of structural colors. For example, if you take a compact disc, you can see the beautiful reflection of the colors from it. And soap bubble, which all of us know. And if you have seen the oil spillage, you can see a beautiful color pattern. And these days, it's also possible to make nail polish, which display more than one color, but it's a non-pigmented. And it's also likely that in future one day we have much more beautiful structural colors in our vehicles. And where these colors come from, these colors or patterns are called structures which are responsible for these colors are photonic crystals. They're also called photonic band gap materials. And photonic crystals are periodic structures which are made up of dielectric materials. I will explain about photonic crystal dielectric materials in the coming slides. And they exhibit some of the interesting property called band gap. And the band gap forbidding the propagation of light of certain frequencies. So I told about the photonic crystals or photonic band gap materials. So what is photonics by itself is a branch of optics or a branch of anything that deals with the light and which control the photons. And as much that it's just like an analogy with the electronics, which depends on the electrons. So if you look at, for example, in conductor, semiconductors, and insulator, and we see a difference in the valency and conducting band. In more detail, if you look at here, the energy regions for which there is no electron state exist. So here, what we see in this case. And that results in what we call the band gap in electronic materials. In the same way in the photonics materials. So what happens in dielectrics? Because there is a certain energy gap, therefore the band below that energy, for example, for if you take metals or insulators, semiconductors, then you have a energy gap which is below 4 electron volt. Then the bands below this energy are completely filled. And of course, with the high temperature, you can excite electrons, which is of course is not true for every materials. And such materials are dielectric or insulators. They're one and the same. What happens for dielectrics? They cannot absorb visible or NIR light. The reason is that the photon energy is not sufficient for transition from valency band to the conduction band. So photon energy becomes sufficient when you have, for example, high energy radiation, such as UV, which causes a very strong absorption. So in photonic crystals, the states of the light field is used in analogies to the states of the electrons. So in the same way, like, for example, the electrons cannot be described with a plain de Broglie wave in a medium with a periodically varying electric potential. The optical plane waves are no solutions for wave equation in medium with a periodically varying refractive index. So it's an analogy with the electrons. We can use the light here. And in analogy with the plane de Broglie waves, we can generate a periodic structure. And instead of electric potential, we can say here the refractive index, which I will come to later. So what happens in the photonic crystals? So if we take a simple example, like here is a pictorial, we have an object, we have light falling on that, and certain light will be transmitted and some will be reflected. And how it happens, and it results in a phenomena called interference. What is interference? If we have now a medium which has one refractive index, then the light passes through another medium which is a different refractive index. So there's a difference in the refractive index of one medium to another medium. So the light will reflect from the boundaries, the upper and lower boundaries of a film. If I have a water, oil, and air has a refractive index of 1, oil has slightly higher, and then water has 1.33. So the oil is in between water and air. So this will result, when you have these two waves which are reflected, this will result what we call constructive. That means there's a phase change between the two waves. That means these two waves can combine. You result in a constructive interference. That means you will see a color. They can also cancel out. For example, if there is a no phase change, then you will see no color because they will not combine, which the resulting is that everything is cancelled. So essentially, if you have an alternative low and high refractive index layers, you are able to generate a pattern that will give you a color. So if there are three different factors that depends, one is the refractive index of the medium, number of the layers, and the thickness of the layers. As I said that if I have a low refractive index, which means the air it is 1, and the higher refractive index medium, like water, so air is 1, water is 1.33, it means that the light travels 1.33 lower compared to, much slower in water compared to air. Then we see a phase change of around 180 degree, then there's a perfect constructive interference. You don't have to be exactly, but the opposite, for example, now it is going from water to air, then we don't see a phase change because we are going from higher refractive index material to lower refractive index material. Then the phase change is 0, then we don't see a color. So the one way is to achieve this kind of interference pattern by regularly placing high and low, high and low, high and low refractive index materials. Here is an example of soap bubble and oil. And in soap bubble, we have a thin air. Then we have film, which is of course a surfactant and water. And then we have air. So the light essentially goes through three different medium: low, high, low. And oil bubbles or emulsion, it's also exactly the same. You have air and then your oil. Oil has higher refractive index than water. So irrespective of, there are three different material, the concept here is the same: low, high, low, low, high, low. So the oil is lower than, no, higher than water. So essentially we see a similar color pattern. The other phenomena that responsible is a diffraction. And diffraction is slightly different than interference. Often it can be misunderstood. And this is essentially due to the bending of the light. It can be a certain defect, or it can be, for example, this what happens in the cloud droplets, and we see scattering of the lights in the sky, and we can see a beautiful pattern. But the diffraction and then the interference have certain differences. As you can see, as you saw, that interference is combination of the two different wavefronts, which eventually originate from the same source. But whereas diffraction is due to the interaction of light coming from different parts of the same wavefront. So this main difference that makes these two phenomena slightly different. So in order to achieve this, whether the color coming from interference or diffraction, we have to create structures or patterns which meet these two criteria. So what are photonic crystals? And first I told these are the phenomena, and this phenomena can happen in structures called photonic crystals. These are nothing but highly periodic structure. They can modulate a refractive index in 1 dimension, 2 dimension, or 3 dimensions. Here is an example for a one-dimensional periodic structure. What is one dimension? You have periodic structures or alternate layer only in 1 dimension. And for 2-dimensional, we have direction like this and also in this direction. And for 3 dimension, we have all 3 dimensions, x, y, z. They're highly periodic in this. Therefore, the structures, depending on that, they can modulate the light or refractive index in a given directions that leads to the structural color. Photon crystals, 1 dimension was already discovered in 1887 by Lord Rayleigh, but the 2-dimension and 3-dimension photonic crystals was first theoretically proposed by Elie Ablonovich in 1987 in a PRL paper, and it is purely a theoretical paper. At the same time, there was also 3-dimensional photonic crystal, which was experimentally and theoretically proposed by Sajeev John. So these two form the foundations of the photonic bandgap materials, the modern photonic bandgap materials. They're also the same phenomena that what we see in optical fiber, whether it's a silica optical fiber or polymer optical fiber. So what about a little bit more detail? So they contain very periodic structure which can modulate the refractive index in a given dimension. So just like I showed about conductors, semiconductors, and insulators, they also have forbidden regions. So it means that light can be dispersed. And these are the photonic band gaps, as I already discussed. And it means that once the light is trapped, you can modulate the light, and you can also propagate the light. And is advantage, such as I said, for example, in optical fibers, where we can trap, manipulate, and propagate light for really long distance, because they are not able to dissipate the light. But nature utilizes some of the simple molecules or materials to generate these kind of structures in a highly efficient manner. And if you look at the nature, irrespective of organism and material, they're all made up of either of these materials. Either chitin, keratin, cellulose, or guanine is an exception for a molecular level photonic material because it has the highest refractive index of 1.83. And then we have reflectin is a protein, and there is an aragonite, is calcium carbonate in sea cells that you can find, and a collagen in mostly in mammals. So it can be one or combination of these materials that leads to the structures. So let's discuss some specific example for photonic structures in nature. And I will discuss about 1-dimensional structure, give 3 different examples for this. We go on to 2-dimensional photonic structures. We give a couple of more examples, not discussing this insect eyes. It's a two-dimensional crystal, but not really photonics. It's for wetting. Then we discuss about three-dimensional photonic crystals and then helicoidal structures and some miscellaneous examples. One-dimensional photonic structures, one of the finest example is a hibiscus. And this particular flower has a white and a purple patch here. And this is not pigmented color. And often we might think that it's a pigment, but surprisingly, it's not a pigmented color. What is it? We have a white patch with a red pigment or reddish or purple, depending on the orientation. And this is iridescence. Just I showed you in the very first slide of the color changing. You can just rotate, take this petal and show it a different angle. You will see a different color. And you can see a blue color, green color, and a yellow color, depending on the angle in which you can view this. And interestingly, it is visible to our eye, because there are numerous flowers where these are not visible to human eye, but insect can recognize this. For example, this kind of color or iridescence can also be seen in many tulips, but unfortunately we are not able to see this unless we separate those structures. And here is a little bit more detailed explanation for this, what actually happens. If you take the scanning electron microscopy of the structure, if you look at the white patch and also the red patch, here is the white patch. What you see is the smooth structures here. And if you look at this red patch, you already see a lot of different colors here. If you look carefully, then what you see is the nicely grated structure. These are diffraction gratings with a specific pattern and a very well defined distance between these stripes. And now you can compare it. Now what it is shown here is that an epoxy is a polymer which was placed on this colored petal. And in the right side here in the B is the same type of experiment done using a compact disc. And then that replica was taken. It is the SCM image of the replica. You see a nice grated pattern, which is similar to actually compact disc. And that's why you can see this color. And here it shows the view from this is the top view. It's the view from the side. And you can compare, for example, here, how the tulip by itself look like. And you have nice, beautiful pattern, but within the pattern, there are extremely smaller pattern that appear. And this gives a iridescent color. And quite differently, it is not following exactly same as, for example, compact disc. In that case, we could see the color that we observe in compact disc, but slightly differently because it is rounded structure, not square or well-defined pattern. In CD, these are highly fabricated, but this is fabricated in the nature using a different phenomena. And the periodicity, as I said, that it should be the order of wavelength. It's about 29 micron. So this is purely made up of cells, which are epidermal cells. And why such colors are important? The colors are important. They have certain biological significance. And one of them is called is for pollinating. And many other functions are unknown. And in this particular paper in Science, they've also done what's called the behavioral study. What's behavioral study is like it's called a differential conditioning. Like taking bumblebees, which are known to pollinate these flowers, and taking those who has never done the pollination, these are called flower naive bumblebees, and trying to experiment their ability to distinguish two similar colors. One is iridescent, one is non-iridescent. And how they have done is just by preparing a synthetic iridescent flower. So that has a similar pattern. And then by taking a replica from a compact disc. And then the disc can be also made colored using pigment, for example. So you have iridescent just by taking the compact disc gratings and a replica, And then same replica can also be colored using a pigment. So you have similar colors, iridescent and non-iridescent. And then what you do is that you give a reward. For iridescent color, which are not pigmented, you give sucrose, a sweet reward for the bumblebee. And for non-iridescent disc, you give a bitter reward, that is cumin hemisulfate salt solution, and which is sour and bitter. So now the idea is that what happens if the bees visit? You leave the bee in a bitter reward, where it will go? Will it go randomly to one place, or will it recognize only iridescent colors, not pigmented color? And this was the example. So what happened is that once they do this condition, then sequentially they removed the bees and they developed the new bees. And what they found, 80% of them end up in all the time the iridescent color, not pigmented color, irrespective of the reward. So if you change the reward, for example, you also give the reward in the colored ones, pigmented colored, same as in the iridescent color, they will always go to the iridescent color. And this is very interesting because they have some unique ability to use iridescence as a cue to pollinate. So that is why the colors are useful. So this kind of structures can be found in tulip, and in tulip you can see much more beautiful grating, very similar to compact disc. And another example is in the insect field. It's called Japanese jewel beetle. Japanese jewel beetle has a beautiful green color when it is at rest. And then you see this nice purple stripe. And between the green and the purple stripe, there is an orange color. And if you turn it upside down, you see the nice reddish orange pattern. And interestingly, if you take all these different colored places and take a cross section and look at high resolution electron microscopy, you don't see many differences here. What you see in all cases, there is a nice a patterned structure. These are one-dimensional layered structure. And there is an alternative layer of high and low electron density. And the difference between these three is that they have different number of layers. The number of layers are different, and also they are not discrete. For example, you can see that they're very nicely observed. There is a nice periodicity, but this periodicity is not very regular. And because it's not regular, they are able to modulate the light and change the color if they want rapidly. So this shows that simply that by changing the same object here, and I think this is made up of melanin, and they can chitin and melanin. So they have different refractive index, they have different electron density, and just by the thickness and the number of layer, you can change the color using exactly the same material. So this saves a lot of energy, resources, and time for materials. Another interesting thing is that Morpho wings, they have very high reflectivity and with a selective wavelength of light. And unlike many other objects like a flower that I discussed, they have this color visible in all angles. So not like in a specific angle. And you can also see there is a difference in the color if you take different types of Morpho butterflies. So how to explain this pattern or differences in the color, ability to see, and the transparency? A simple multilayer like I showed for Japanese jewel beetle can explain like why we have high reflectivity and why there is a uniform color and variation of color. Only reflectivity can be explained, but we cannot explain 2 or 3, which means that uniform coloration or variation of color among species. And if you look at the high resolution electron microscopy of this cross section of the wings, what you see is that they have some specific pattern, and this kind of alternate pattern, and this alternative patterns which are not having two different material. Instead, what they have is the air cavity, and that is why it is you can have very wide range of very broad angle vision of color for morpho butterflies. And the only difference here is the size and the number of these graded layers. And this as simple as that. Another interesting example that we can see is in the fish and beetles. And these are examples for physiologically controlled color. And it means that it has nothing to do with a well-ordered, it has to do with a well-ordered structure, but it's not a permanent structure. A structure that, depending on the environment, the animals can excrete certain body fluid that will modulate the color, essentially the refractive index. And in both cases, in the fish and beetle, what they have, you have a layer of guanine crystals, the molecular level building block, which forms a nice microcrystals, an alternative layer of guanine crystal and cytoplasm. Now, guanine has 1.83 refractive index, where cytoplasm is close to water, around 1.4. And so this alternative layer, when, for example, this beetle was attacked by a predator, or you touch it, you will see the color change immediately to red within few seconds. This is because it is able to excrete some body fluid that will fill this cavity or change the refractive index that eventually change the color of this object. This is a dynamic and physiologically controlled color. So what we have seen is one-dimensional photonic materials, which can come in a simple diffraction gratings, layered structure, or this graded pattern. So then what about two-dimensional photonic structure? Two-dimensional photonic structures are not very common, and generally they are used for some other functions such as wetting and superhydrophobicity, like in moth or butterfly's eyes. But there are a few examples. One of the example is this sea mouse, which was identified in 2001. And the sea mouse has a beautiful color pattern, and this color pattern can be visible, and you can also see, depending on the angle, different colors. It can be, when you see from one angle, it can be bright red or greenish. And the reason for this is that it has a two-dimensional photonic crystals, and these fibers, if you take the cross section of this, you can see very nice hollow cylindrical structures arranged in a two-dimensional pattern here. And what they're made up of, they're made up of, again, simple building blocks such as, for example, chitin. And if you take one fiber of that, one hair, and then you look at, count the number of these hollow cylinders, you can see that at the center there are big, and as you move towards the edge, you start to see the size decreases. And this is made up of chitin, alpha chitin, which has a refractive index of 1.4. And then what you have at the center, these are hollow. So there is a difference in the refractive index between um, chitin and then the air. So they're able to trap and propagate the light and also modulate the refractive index in two dimensions. So it's both X and Y. In the previously, we had only in one dimension layered structures, but now these are in both the directions are used for patterning these kind of biological structures. And another example that I showed in the beginning is peacock feather. Peacock feather is even more interesting. If you take, for example, the stem, here is the stem, the main structure, and then you have the branching from the stem are called barbs, and the small hairy structures are called barbules. Now, if you want to look at the green and brown color, here, for example, is a scanning electron microscope is showing the green color responsible structure. And here, the structure they're responsible for brown color. And what is the difference between this? So it has a core of about 3 micrometer, yeah? And the barbules contains array of two-dimensional array of these rod-like structures. And these rod-like structures are arranged in green as a square lattice, whereas in brown, it is a rectangular lattice. So it's exactly the same material, but only the difference is the lattice and the way they are arranged. So that can generate different color. And this is made up of melanin. These rods are made up of melanin, but they are interconnected using keratin. So what you see is these are the voids and the rods in between, and the rods create a beautiful pattern of square two-dimensional crystals or rectangularly arranged two-dimensional crystals. And exactly the same material, same size, but only slightly they differ. In each color is slightly differ in the lattice pattern. One other fascinating example is bird of paradise. You might have seen this if you have seen David Attenborough's Planet Earth videos. And this bird of paradise has this nice dancing moves, which is called ballerina dance. And if you observe carefully here in this dance, the most important is that the structural color that observed, the pattern that arises here. It is a beautiful shiny silver shiny black feather, but when it starts to dance and attract a female, then you can see the color pattern appear here and also behind his head. And here is the female. The female bird is a dull brown color, and it has not evolved anything much in thousands of years. But whereas the male has adopted a beautiful structural color. And what are these colors made up of? And this is very interesting because it shows a three-directional reflection. The color can change nicely depending on the angle that view. And these colors, if you take now the silver-like feathers, and also if you take a look at these greenish shiny colors, you see they are made up of similar materials or exactly same melanin, which I discussed, for example, octopus ink, where there's simply polymer. But here they are arranged into very well organized structure of certain size. And in this case, for this, it's about 250 nanometer, and they form nice layers, two-dimensional layers, and the spacing between the layer is about 400 nanometer. And now what is the difference between these two colors? Here, they're slightly different because the size of this particle, melanin particle, is smaller. At the same time, the distance is almost half that of the silver color. So just by tuning both the peacock feather and this, the size and the spacing between these rods, you can now modulate the light and create different colors. And most important that these colors are very stable. Unlike pigmented color, if there is no dietary supplement or it can undergo photo bleaching, these colors are stable. They can stay stable even forever because it's purely based on the material. Only once the material decomposes, this color will disappear. Otherwise, the color will retain. So there are beautiful melanins that also helps to filter and generate and separate certain colors. And how it modulates this color is still not yet known exactly. It's able to generate so many different colors just with a span of time. We go on now a little bit about 3D photonic crystals as the next step. And as I said, then for 2D and one-dimensional, especially for one-dimensional, there is huge number of examples. I'm showing only limited number of examples just as a representative ones. 3D photonic crystals are found largely in beetles. And many beetles, generally they display shiny colors and which you cannot see fully colored beetles in the night, for example. But there are certain beetles which has a uniform metallic color. When you see from any direction, you see this uniform color. And these are colors which are very similar to, for example, opals. And this color is due to the presence of certain structures on these small scales. And these scales are present about a millimeter in size. They are present in this hemispherical body of these beetles. So what is it made up of? If you take a cross section of these small patches, what you see is a three-dimensional organization of these spherical nanostructures here. And the lattice constant of this will approach, for example, the wavelength of light. If you take a reflectance spectra, then you can see that it nicely matches and it's around 550 nanometer. So it is very common that you can see this kind of structures in opals and many other structures in the nature, and insects follow exactly the same principle. Another beautiful example is butterflies. This particular butterfly has many different color here. The black, green, the red patch. And the large green color is always dominated in the forewings. And then you have wings with the red patches here. And if you take now the cross section of this, look at the electron microscopy, you can see that the colors are very similar, for example, opals. And there are two different structures now you see here. One is these are grated structures, which are in between these two grated structure. What we have is a three-dimensional gyroid-like structure, a very complex network like this. And these are made up of chitin, which are filled with 30 to 40% of this. And interestingly, it's very important to know that in many beetles, and we don't have, for example, simple cubic type of structures. I mean, like in butterflies, we don't have a simple cubic structures, very complex gyroid type structures. And in beetles, you can see cubic structures, as I shown in the previous example before this butterfly. But in butterflies, you see very complex gyroid structures. So this is very interesting that these two cannot have similar structures, but they can display similar colors also. And there is something has to do with the evolution and maybe there are structures which has not been identified yet, but it's very interesting for those who study evolution and the color and the significant. It can also be so that, for example, to create a gyroid-like structure requires much more energy or much more efficient construction of the structures, whereas beetles may not be able to construct them in the same way as, for example, butterflies. So what does it do? And this color change is very interesting because they can escape from predators because of this, because they can quickly adopt this green color, which can match, for example, of green leaves, and they can change the color at any time. This allows them a quick camouflage, which means that color range. So the final part is the miscellaneous structures. One of them is the helicoid structures and polarized vision. We cannot have polarized vision, but many insects can selectively perceive, navigate, and communicate the one type of circularly polarized light. And this is due to the structures which are not highly periodic, as I've shown in the previous example. This is totally a different class called helicoid structure. And the helicoid structures can form very beautiful angle-dependent reflection, and they can selectively visualize one type of polarized light. And an example here is a beetle. This has a shiny metallic color, and it can selectively reflect left circularly polarized light. And now here I show is the under a normal. It contains both right and left circularly polarized light shined on the beetle. Then you see this color. Now if you block left circularly polarized light from the beetle, then it will lose its color. So this is rather interesting for developing many materials which can selectively, we can make selective vision of polarized light.
So it is because of the presence of some of the chiral and helical structures that are generally made up of either chitin or um, cellulose, which are rod-like cellulose particles, and they arranged in this kind of pattern, which undergoes the helical pattern. It's a normal line here, and this is going in a helical pattern. There is a layered structure, and as you can see, it has a certain pattern. This pattern, you can say that the pitching. So this pitch height determines the color that you can see. And this is not only in the insect, but you can see in many fruits, because fruits are essentially made up of many of these are made up of cellulose. If you look at this color, this is not pigmented color. That's why you can store this fruit for 1,000 years. The color will not fade. And if you take the cross section, you look at the SCM image, then you can see these helicoid structures, nicely arranged of cellulose. More detailed example is here. This is a fruit called Folia condensata. This is used, for example, as a decorative material, because it will never lose its color. And again, it's not because of pigment, it's again because of the cellulose structures, which are arranged in a helical pattern. And now you can calculate the color, why it gives, for example, shiny color, blue coloration, because as I said, it depends on the pitch height, like how many layers or distance it takes to complete one full turn or 360-degree. And in this case, the pitch length is P is about 290 nanometer. Now there's an equation where you can substitute this value and you get the wavelength around 450 nanometer. That 450 nanometer correspond to the same color in the spectrum of the visible spectrum of. So interestingly, this is very interesting because it's a high reflectivity, and it's a record that there is no other biological molecule that shows structure that shows so much of reflectivity. And a combination of diffraction and interference can also be found. This is a two-dimensional, again, material, not three-dimensional, but rather different from any other category. The mother of pearl. You can see beautiful but very diffuse color. Now, if you look at the surface of this, you will see this kind of not very well patterned grooves. And then if you go on, take a cross section of this, you see even smaller structure of calcium carbonate, which have about 5% other organic polymer, like maybe chitin or some proteins, and which are between these crystals. So one of them result in what we call multi-layered interference. The other one gives a diffraction. A combination of diffraction and interference gives you this diffuse color. And finally, what we can discuss is not everything is interesting when it's colored. And color is important, but also it requires a lot of time, and it also takes a lot of materials and energy from the organism to create. There are certain organisms that can also have non-color or bright colors. They scatter light or they scatter white light. But to generate white light, it requires some effort. It's also not easy. And it's difficult because there should be a uniform, they have to reflect the light in the same efficiency. And these are some rare example of beetles. And such a ability is not possible by many organisms. So one of the examples is a special case here is butterflies. You can see that they have nice white light, white coloration, bright white coloration. And this is because of the pigment that they have. And these pigments are called terins, which are different chemical components which result in certain number of pigments that selectively absorb certain light. And for example, if you take leucoterin, it can absorb in UV wavelengths. And this one, because of the higher conjugation, more functional group, this can absorb at 500 nanometer. And here is the same, two different butterflies. The one is the white, bright
coloration, the other one has a multiple color, and here there is a black patch in this white wing. Now, if you take the white portion of this electron microscopy, you look at these stripes and you look even higher magnification of this, you see nice this pattern. The pattern, you can see that ellipsoidal objects. They are made up of these terrene pigments, which self-assemble into nanostructures. And if you take the black patch now, the D, which shows you the SEM image, it's not color, it's a black color, but there are no this kind of ellipsoidal patches. Same holds true for also this particular species, where it's color, you take any portion of this, you don't see these ellipsoidal structures. So it eventually has to deal with this kind of nanostructures that helps you to have a very bright coloration. And in this case, it is due to multiple scattering, and it is essentially made up of very low refractive index materials. And another important phenomena why you can see white coloration is because if you take the structure scanning electron microscopy of this, you see highly anisotropic, means not spherical structures. These kind of small structures, these structures, if you look even more further, you see highly disordered structure. The disordered structures are the one which are responsible for such a bright light coloration. Yes. And a little bit more, the disorder, how disorder helps is that it helps us to avoid any spatial correlation. It fills the space, but there is a lot of void in between them. And you can create this kind of structure simply by following this principle. And this is why many insects are able to do this. And we can actually change the, for example, if you have highly transparent material where complete light transfers, you can change, for example, the order in that, and it can turn bright. And such examples are known in literature. So not everything is colored, not everything is white, but there are also transparent materials, and that's called biological transparency. And biological transparency is mostly seen in objects which are present in the sea or in the water. Because for that to happen, you have to have materials where the refractive index will match close to that of water so that these are invisible. And but there are challenges, right? The challenge is that if you want to have a complete transparency, the entire body has to be transparent. Only then it is invisible to the predators. And except the eyes, if you see many of the transparent animals, only the eye is not transparent. That's of course not possible. So, but biological transparency is also a challenge because they want to avoid the predators, but they can lose their transparency very quickly. Here is, for example, a type of shrimp which is highly transparent, but if you take it to add a very small change, like parts per trillion of salt, then it loses its transparency and it becomes opaque. Immediately, it will adopt and it will become opaque, and the predator can easily identify. So, if you slightly change 1 or 2 degrees Celsius, the transparency from 40% to 0%, it will change rapidly. And this is not good. So, this is also difficult to maintain. So what they do in such streams, they do some behavior adaptation. That means that they find a way to overcome this. One way to overcome this is that to go to a place where there are like not much change in the salinity or temperature. It's always true that the surface water is highly sensitive to salinity and temperature. You go deep water, then you have lesser possibility of change in this. So they have adopted this kind of behavior adaptation. So I would like to conclude by saying that we have seen in this webinar, I tried to go through, cover many topics, but only idea is that nature can modulate light, nature can interact with the light, and they can manipulate light. And we can find many examples for structures like 1-dimensional, 2-dimensional, 3-dimensional structures. There are some miscellaneous structures that are example that leads to what we call as polarized vision, and then the scattering and the transparent structures. And I hope that many of you now start looking at structure and see, oh, is it a structural color or pigmented color? And I'll be very happy if you get inspired by some of these examples. And I'll be happy